All right. Hello, everybody. I hope everyone's doing well. It is June something. Tonight, it's 9 o'clock, 8:30, something like that. I'm recording this video at night, so I'm using some lights. I don't normally do it this way. All semester long, I've been having the windows open and using natural light, but trying to get down near the end, and I've got my wife working from home and my kids, and so I'm running out of time. So I'm doing the best I can. So you may hear screaming and crying. Some of that might be coming from me, but I figure at this point we're doing the best we can. So last lecture of the semester, and here we go. We're going to talk about neurons today, but I wanted to get this going and up so you had more time to study it, just because it's a nice thing to do. So anyway, we're going to talk about the nervous system, and for some reason, I guess I went one slide ahead. I should have started here. Anyway, starting with all the way back to Cnidarians, all these animals have a nervous system with varying degrees of complexity, and we'll get into that a little bit. But let's first start with a very important cell that is involved with the nervous system, which is the single cell called a neuron. And there are some basic parts to a neuron. So this again is a single cell, and neurons are unusually shaped cells. So they can be very, very long. You might have one neuron that goes out of your back and goes all the way down to your foot, for example. And that's a single cell. And then you'll have a bunch of them bundled together that will form like a nerve. But in fact, that is still one individual cell inside there, in which part of it can be very, very long. So typically what we talk about is this end of the neuron is called the dendrites. That's the receptive end of the neuron. We'll get to that in a second. This part of the neuron is called the cell body, or the soma. And it's where the nucleus of the cell is. And then there's this little tapering part that comes down like this. That's called the axon hillock. And then there's this one part here that's usually pretty long that goes to a particular location, and that is the axon. So this is one generalized type of neuron. There are, in fact, lots of different neurons with all kinds of shapes, but this is the one we'll talk about here. And then the neuron goes down and it ends somewhere, and it can end on either another neuron or on a muscle or on a gland. Okay, now the first thing we need to do is talk about how the neuron works. And if you were to take a voltmeter, like the kind of one, where you measure the electricity in the wall or on a battery, and you have two probes that you would stick on a battery or in the wall, you could stick one inside and one on the outside. You could compare the difference. And they actually do this. Lots of people that study neuroscience do this on different animals. Sea slugs is one of them. I had a friend in grad school that studied crickets this way, where they measure neuron pulses in individual neurons, like in the brain of a cricket. And anyway, if you measure the voltage there, you would find that when the neuron is not firing, it's just at resting state. It's what we call at resting potential, and a neuron is usually at around minus 70 millivolts or so. So it's negatively charged. And the way it's negatively charged, the way it gets there, is for the fact that there's a couple of different proteins that are involved with the transport of sodium and potassium in and out of the neuron. And I always try to remember to mention this, but they show you like one picture here, and it shows you potassium and sodium and then this sodium potassium pump. But these really go all along the neuron. So they're located all throughout the neuron. And in fact, the black line that you see there is actually the cell membrane of the neuron. So if you were to follow that black line, you would have scattered along there something that looked kind of like that, the phospholipid bilayer. I didn't mean to do that one. That was a mistake. But anyway, you'd have the phospholipid bilayer there. So you have a cell membrane there. And you have a couple of different proteins that I'll get into in a second. But first, let's talk about this one, because this is the key one right here. This is a sodium-potassium pump. And it does exactly what it sounds like. It pumps sodium and potassium. So what it does is it pumps 3 sodiums out, and for every 2 potassiums, it pumps in. And both sodium and potassium are positively charged as ions. So, and this is an active transport pump, so it's using energy to do that. You're pumping 3 sodiums out, 2 potassiums in. Now, in addition to the sodium potassium pump, you have these channel proteins that can open and close, that can be sensitive to letting sodium or potassium move through the cell also. And what we find is that the sodium one in the resting potential there is usually closed. Okay, and the potassium one is a little bit leaky. So some of the potassium can leak out. So what ends up happening is you're pumping 3 positive ions out for 2 positive ions in, and that itself is going to make the cell more negative because you're putting more positive things out than you're bringing in. And then as you make that concentration gradient, you're also then leaking out the potassium due to the gradient difference, because there's more in here, it leaks out, and then the sodium channel is closed. So as you do that, you end up with more positive ions on the outside than on the inside, and so you have a cell that's then slightly negative. Now, the way a neuron works, the way neurons function, is they're what we call all or none action potential. So the neuron either fires an action potential or it does not. So if you're at minus 70 millivolts here and you let a little bit of sodium in or potassium out or whatever, this will kind of move around a little bit, but very little happens. It sort of resets itself. So there's a threshold point that you have to get to. And once you hit the threshold point, once you get up to, and it's different for every neuron, for different neurons, but if this is minus 70, if this is minus 70, maybe this is minus 45 or something like that. Anyway, so as you get more positive, once you hit a certain point, what happens is the sodium channels then open up. So there are what are called voltage gated sodium channels. And the voltage gated sodium channels open when you hit that threshold. And because you pumped so much sodium out and the inside of the neuron is negative, you have sodium being drawn in. It's rushing into the cell. It's not only at the concentration gradient difference, but it's also electrically pulled into the cell. And so very, very quickly, in a matter of milliseconds, you get this action potential, and then it resets and you end up back where you were. So in the resting state, in the very beginning, what you see here, number 1, you have both sodium and potassium activation gates are closed. The interior of the cell is slightly negative, and it turns out that sodium rarely leaks out, but the potassium leaks out a little bit, which sort of helps the cause, okay? Then during depolarization, that's number 2, you see here, the sodium activation gates are open on some of the channels, and that allows some of the sodium to come in, and the interior starts to become more positive. It's still negatively charged because it starts as minus 70. So if it goes to minus, say, 45, it is more positive than it was before, but still negatively charged. Now, once you get to number 3 and you get to a point where you have enough sodium in there, all of a sudden the other sodium channels open. That's what I was talking about, the voltage gated sodium channels. Those open up, sodium comes rushing into the cell. The potassium activation gates are still closed, and that's the action potential. And then what happens is the inactivation gates on sodium channels end up being closed. So they open and then they close really quick. Activation gates on potassium channels then open, and the cell starts to go back to being negative again. And in fact, it goes just a little bit. We call it the undershoot that you see there. The sodium channels are closed, potassium channels start to close, and the membrane goes back to its potential. But there's a little blip, a little undershoot, and then you're back at resting potential. All that again measured in milliseconds. Now, what ends up happening is an action potential is a localized event, and it starts in one place and it travels down one direction. So it goes typically from the dendrite down the axon to the synaptic terminals on the other end. But there's this pause as it's resetting over here, so it can't really flow back this way. So neurons usually fire sort of one direction, and that is due to what we call the refractory period. There's this moment where it has to reset, and until it resets, it can't fire again. So the neuron can only fire one direction, and it can only do it so quick, which is still measured in milliseconds. Now, what we talked about so far is one neuron firing, going from the dendrite in to the synaptic terminal. So if we had a neuron, this is all we've done so far is we send the electrical signal from one end to the other end inside a cell. But the real magic starts to happen down here. So this is a chemical synapse. So when you get down here, what happens is the electrical signal comes down here as it goes from resting potential to action potential. And then once the action potential reaches the end, the synapse or the synaptic terminal on this end, these voltage gated calcium channels open, and that causes these vesicles here to fuse to the membrane and leak out their neurotransmitters. The neurotransmitters then bind on the other cell that you see here, and then those can relay that message from neuron number 1 to neuron number 2. And there are a bunch of different kinds of neurotransmitters that do different things. So acetylcholine, for example, tends to be excitatory in vertebrate skeletal muscle. You don't need to memorize the structures of them here. Then we have norepinephrine. That one can be excitatory or inhibitory. We'll get to what that means in a second. This one here, dopamine. So there's a couple really big ones that are very significant. And there's over 200. So we'll just name a few that we can talk about here. GABA, serotonin, dopamine, and norepinephrine. So dopamine, for example, dopamine's been associated with Parkinson's disease. So low levels of dopamine tend to be related to Parkinson's disease. Some neurotransmitters, there are some drugs that can help increase the release of certain neurotransmitters, and then there's some that we have a hard time doing. Dopamine's one of those that we've had less success with. It turns out high levels of dopamine are associated with patients that have schizophrenia. The mental, what am I trying to say? The mental health condition where people sometimes often have trouble and confuse what's real and what's not. And sometimes have hallucinations about things or hear voices or see things. And there's a variety of ways that comes out. Anyway, dopamine's also been found to be important in things like sleep and mood and learning. And many times what ends up happening is it's the relative ratio of something. So for example, when you exercise tends to lead to an inhibition of GABA, but it increases in dopamine, and that combination is often referred to as what people call the runner's high. People that do aerobic exercise, running is one of those, tend to get this sort of mood swing that is very sort of beneficial. It's a beneficial mood swing. Mood swing isn't normally something you hear of in a good way, but this is a very calming kind of effect due to, they think, this inhibition of GABA, but an increase in dopamine. Then you have serotonin. Serotonin is the famous neurotransmitter associated with depression. People with low levels of serotonin are thought to, are definitely associated with depression, and there are a lot of medications on the medications that people can take that are designed to affect serotonin levels in different ways. Okay, so serotonin is generally an inhibitory one, and again, we'll get to that in a second. They've also found that low levels of serotonin not only associated with depression, but also with aggression. And it turns out someone did a study on serial killers. I don't know how you find that many serial killers, but anyway, low levels of serotonin, maybe they're depressed because they're serial killers, or I don't know. But anyway, study done at least suggests that serial killers seem to have a low level of serotonin. Anyway, now what happens, I mentioned the inhibitory and the excitatory part, is that when you have a neuron that fires and sends the action potential, it depends on what that neuron does. So when I have one neuron here and I have an excitatory neuron, that's sending this impulse to this neuron. And if it's excitatory, it would increase the voltage in it. It moves it more towards threshold, but sometimes one neuron going to it isn't enough. So what can happen is if you had E1 here, for example, the E neuron could fire really quick, two times really quick. They call that temporal summation, and the firing of those two neurons really quick can stimulate this neuron to fire. Or what you could have is spatial summation, where you have excitatory neuron 1 and 2. Those both fire kind of near each other, and the combined effect gets you to threshold, like you see there. But you could also have an excitatory neuron and a inhibitory neuron. So the excitatory one fires and the inhibitory one fires, so you don't get it threshold. And so the reason that's kind of important is if you think about neurons, they either fire or they don't fire. So they're like a light switch, and the switch is either on or off. And so the way our brains work and our nervous system works has to do with turning on and turning off series of neurons. So for example, when you're listening to this right now and hearing my voice and seeing me or whatever, you're firing certain pathways of neurons in your brain that allow you to learn or not learn this information. And when you study or you practice, what you do is you start to reinforce how some of these neurons can bind with each other from neuron 1 to 2, and you can practice. As you practice, you can get better at getting those neuron pathways to fire. When you try to think of somebody's name, or you try to recall the name of a movie, the name of the movie is encoded in your brain in a series of neurons that are turned on. If you can't turn those neurons on, you can't get to that name. And so that's kind of how it works. And it seems kind of weird that these on and off switches can code for all that information and all those memories and thoughts and all that in your brain. But that's how it works. There's billions of these neurons that are doing that. And that's kind of the same thing. The way a computer works, in the sense, is that it's ones and zeros. And so the entire computer, think about everything your computer can do, all that's coded for in one way or another in ones and zeros. And that's what's happening here. So you can get with what we call postsynaptic potentials, what happens after the synapse. You can get temporal summation, where again you have two signals that don't reach threshold, but in time, they do it quick enough. Or spatial, 2 signals set off about the same time coming from different spots. And you can get temporal summation, spatial summation, or you can get inhibition, which is the next one. So the next thing we're going to do is talk about the vertebrate nervous system a little bit. We'll talk a little bit about brains, the different parts of the brains. This picture shows you some of the different brains of the animals that we've looked at or would have looked at in lab. And before we go into looking at the brains and the parts of the brain, we want to talk about the peripheral part of the nervous system. So the central nervous system is the brain and the spinal cord. That's the central part. And then things go out from there and communicate with the rest of the body. And that is your somatic nervous system and your autonomic nervous system. The somatic nervous system is what goes to your muscles to make them move. And the way you work out your muscles in the gym or whatever you do is by making your peripheral nervous system, your brain, going from the peripheral nervous system to the somatic nervous system to cause those muscles to contract. But first, let's talk about the autonomic part, the automatic part. And you have parts that control your digestive tract that you don't worry about, the enteric system. It sort of does it on its own based on what your brain's telling it to do. And then you have these 2 very interesting parts, the sympathetic and parasympathetic parts. And we'll go into those here in a second. So autonomic, again, usually regulates the internal environment. It's usually involuntary. And then somatic, like I said, usually voluntary muscles that you can move. And sympathetic is the one side that you don't typically actively control with your mind, that is often referred to as the flight or fight response. And then you have the parasympathetic division, which is the rest or digest response. Now, this is one of the charts you're going to need to know about what happens in the sympathetic versus parasympathetic. So remember, sympathetic is fight or flight, okay? And this one's rest and digest, okay? So what happens is this. This looks like an overwhelming chart with lots of things you got to remember, but most of it is pretty easy if you think about it in the context of what's happening. So we'll start with the fight or flight, okay? And you don't need to remember where it comes out of the nervous system, but all along your spinal cord are neurons that relate to these functions, okay? So first, sympathetic nervous system, fight or flight. You're walking out to your car, it's a pandemic, there's riots, there's peaceful protesters, there's murder hornets, there's, who knows? By the time I come up with this, I heard there's mosquitoes now with St. Louis virus and maybe West Nile virus, who knows at this point, right? It's all going down. Nothing surprises me now. Anyway, fight or flight. And so you're going out to your car, these things are all going on, and it's your friend, they jump out and scare the crap out of you. But in doing so, when they do that, your nervous system gets ready to either run or to fight or do both. And so certain things happen. So first of all, your pupils will dilate. Dilate means they get bigger. Getting bigger allows more light in so you can see in lower levels of light what's going on. You're going to inhibit your salivary gland secretion. Salivary glands are used for digestion, and when you're about to get killed by somebody, you think at least they're jumping out to scare you, you don't need to digest your food anymore. Like, you shut that down, okay? Now, relaxes your bronchi. Relaxing seems like the opposite of what you would be doing in fight or flight, but the bronchi are tubes that go to your lungs. And when you relax them, they get bigger. And when they get bigger, you can get more air in and out of your lungs. And therefore, you can get more ATP and you can get more energy for the fight. Your heart rate's going to jump up. That also will circulate your blood quicker. You're going to inhibit your stomach and your intestines, and you're going to inhibit the activity of the pancreas. You don't need to digest things. Now, you're going to stimulate glucose release from the liver, and you're going to inhibit the gallbladder. Now, glucose is a quick, very quick energy kind of sugar, right? Simple sugar. And if you've ever ate a donut, which I used to do, but now I feel terrible, when you do it. Anyway, you have this sudden crazy high sugar rush that lasts for a few minutes and you feel terrible. And that is the sugar crash, which is not good. That happened to me today with my son. He ate his ice cream part first and didn't eat his lunch, and then he became a maniac, and then he felt bad. And then you know what he did? Oh gosh, he ate more of the sugar, like, why would you do that? We just talked about how bad that was. So anyway, there you go. You're going to stimulate the adrenal medulla. Adrenal medulla is going to release adrenaline. And adrenaline gives you that pumped up feeling where you got you're not only your heart rate goes up, your blood pressure goes up, but people get, insane levels of energy and can do things like move cars when they couldn't. You ever seen those shows where someone, a mom's kid's trapped in a car or something, and she moves it like an inch or something like that, because you get this sudden crazy burst of power in some ways that you normally wouldn't have. You don't even think about it. It just sort of happens. Okay, you're going to also inhibit emptying the bladder. Peeing is like a luxury that you do when you relax. When someone's going to kill you, you don't worry about your bladder anymore. Now, the only one where I'm like, okay, you got to think about this one. The parasympathetic division is involved with promoting erection of the genitalia, but the sympathetic nervous system promotes ejaculation and vaginal contractions. So I don't know why exactly. I'm sure there's a good story, but it doesn't fit with all my other parts of fight or flight. Like someone jumping out, trying to kill you, and you're going to ejaculate. It kind of messes up the story. So what I do is I say, my story fits all those. And then when I try to memorize it, I'm like, okay, this one I got to memorize, because it doesn't fit. So if you go on the other side, parasympathetic, it's all the opposite of that. This is like you just ate Thanksgiving dinner and now you're going to lay on the couch and relax. It's all the opposite of the other things. You're going to digest your food, slow your heart rate down, and all that kind of stuff. Okay? Now, the next part, we'll talk a little bit about the brain as the main unit that processes and houses all these neurons that leave the body and control different parts of the body. So we'll talk about the different parts. And first we'll talk about the medulla oblongata and the pons, which are located down here, okay? In the very low portion of the brain, the medulla oblongata and the pons. And those control things like breathing, heart rate, and digestion. Although you can regulate your breathing, you can think about it and breathe deeper if you want. If you don't think about breathing, you don't have to do it because your brain will do it for you. Usually once I mention that you can think about breathing, you usually start thinking about your breathing and it kind of messes it up. You actually breathe kind of weird and it's not right. But if you stop thinking about it, you'll go back to breathing normally. But it also controls heart rate and digestion. Now, the thing that's important there is these are some very primitive, they're very, very basically biologically necessary. So I tell you that because if you're doing something dangerous, jumping off a cliff diving, horseback riding, or you fall off. Anyway, ways you can break your neck, which are not good. This little area, oops, where'd my pin go? Pin. Anyway, this little area in here from there to like there, so you're talking like, 4 or 5 centimeters in there, somewhere in there. Anyway, breaking your neck in those areas has very, very different effects. So people break their neck, for example, and are paralyzed from, say, the shoulders down or from the neck down. But as you go up, that gets increasingly worse. So you get to where you can't breathe on your own, or you need help with digestion, or you can't control your heart rate or blood pressure, all those kind of things. Okay. The next is the cerebellum, which sits down here. It's not really part of the brain stem, which is the lower portion here. It sits in the back. It's its own part. The cerebellum controls coordinated body movements. And so this is often what people talk about when they talk about muscle memory. If you've ever done martial arts or dancing or that kind of thing, or even riding a bike, once you kind of learn it, the cerebellum kind of remembers how to behave while you're doing that particular thing. A good example that I often use is when you're going upstairs. So when you go upstairs or downstairs, the stairs by code have to be kind of a certain height. And so when you walk up the stairs, you see the stairs, you have to take a step with another part of your brain to reach up and get on the step. But then once you start walking, your cerebellum can kind of regulate that repeated movement of the steps. And you can go up the stairs or down the stairs. And it allows you to do dumb things like text while you're walking up and down stairs, which you should not do. But it allows you to do that. And we've all done that thing probably where we thought there was one more step, or there was one step there and it wasn't. And you kind of step and your leg kind of locks, or it does something weird. And that's your cerebellum predicting either another step's there or there isn't one, and you responded funny to that. So just as an example, another example of that, when I was growing up as a kid, my parents, they added on to their house, and my dad, who's an engineer, wanted to do his own stairs for whatever reason, wanted to build his own stairs. And he's an electrical engineer, so he's not like a civil engineer per se, but he did the math and he built these stairs. And anyway, he did a pretty good job. But when you go up, the last step was off by like an inch or something like that. Especially with these floors. So my mom wanted these really big real thick wood floors. So inspector comes out and it doesn't pass inspection. My dad's like, why doesn't it pass inspection? You get to all these stairs out because the last step, that one step is, I can't remember, 1 inch too high, too low. I guess it was 1 inch too high. So what my dad did is he took all the floors out, but he left the stairs in, called the inspector back, and then it passed because with no floor there, it was, at the right height. And then when the inspector left, he put the floors back in. So all growing up, we had to know that last step when you're going up the stairs in my parents' house, that last step, your cerebellum isn't going to take it well, and you're going to miss. So you would trip, you'd have to consciously take a really big step to get over it. But anyway, then we have the midbrain, and the midbrain integrates, receives information, and projects it to the forebrain, which we'll get to here in a minute. And then the forebrain is this part right in here, and that is going to include the epithalamus, which includes the pineal gland, and the pineal gland sits back here. The pineal gland is why none of us can sleep really well at night. Right now, it releases melatonin. And as you're inside all day and you don't know what time it is anymore, or you have jet lag because you traveled somewhere, the pineal gland releases melatonin, and that helps regulate your sleep and wake cycles. And so many of us are all messed up right now. I've been going to bed at 3 or 4 in the morning, and then waking up at 5 or 6 because that's when the sun comes up, and then and then you don't feel good. So, but anyway, what are you going to do but complain about it like I do right now? Anyway, then we have the thalamus, and that's going to conduct information to specific parts of the cerebrum, which we'll get to. And then we have the hypothalamus, which is right here. And the hypothalamus mainly controls this gland called the pituitary gland. And the pituitary gland secretes all kinds of hormones that do things like regulate body temperature, hunger, thirst, sex response, and in part circadian rhythm. Then we have the biggest part of the brain, which is the cerebrum, which is this part here. That's associated with higher thinking. That the cerebrum is really what sets humans apart from other animals because of all the different things it can do. So this is a nice generalized map of the cerebrum. So this is the major portion of the brain that you see here. Now, for example, this region right here, and so right now the eyeballs would be looking this way. Let's say there's your eyeballs, here's your nice eyelashes, and there's your eyeball, and it goes in like that, and you're looking this way. And this is your back. So there's your spinal cord, this is your back, this is your face. There you go, like that. Okay, so the primary motor cortex all along this part of the brain controls movement of different parts of your body. We'll get to that in a second. I'll show you. And then right behind it is where all the sensation kind of those same body parts goes. When you speak or you generate speech, you try to say words, for example, there are two different speech areas that you can see right here. And so when you generate words and you talk, these regions of your brain, those neurons are being used, and there's different ways they can show that in different kinds of imagery where they're doing PET scans and CAT scans and those kind of things. But when you do brain surgery and you stimulate those regions, you can get those kinds of processes to occur. Taste is processed there. Reading is processed in the back. The back portion is related to vision. So if you were to get hit, for example, in the back of the head by an object and you have damage to this region here, you could go temporarily blind or permanently blind. If you had a stroke that affected this portion of the brain, you could be blind from that. Hearing is on the sides over here. So this is on the left side, and then your sense of smell is right below the speech area and sort of in front of the hearing portion of the brain. This figure, you don't need to know this figure. I just wanted to show you this one part right here. But remember, I was telling you about the primary motor cortex there and the somatosensory cortex there. These parts of your body are what are moving when you activate those regions, and you notice kind of right behind them. So for example, your hip, if you try to move your hip, that portion of your brain's where the neurons fire that move your hip. And then if someone pokes you in the hip, you feel it there, for example. You feel it in your hip, but that portion of the brain processes that. So if there's damage to those regions, you either can't move that or can't feel that, or both of those. A little bit about memory really quick. When you talk about memory, memory comes in two forms: short-term memory and long-term memory. Short-term memory are things that are usually stored in the frontal lobe up here. So when you hear something really quick and you see a license plate, or you see someone run out of a store, or someone smiles, or you hear a number, it usually goes in one ear and out the other, and it's there for a little bit. And so our short-term memory isn't very good. Most people can remember 6 or 7 or 8 things in short-term memory that come at you within a few seconds, but that's about it. So in order to do well in Bio 2 and in all your other classes, what you need to do is you need to get things into your long-term memory. And there are different ways to do that. But when you get things in long-term memory, you don't just activate the frontal lobes, you start to activate the portion of the brain called the amygdala and another portion of the brain called the hippocampus. And these frontal lobes interact with these regions, the hippocampus and the amygdala, to help consolidate those memories and make them stronger and reinforce them so you can recall them later. Interestingly, these areas of the brain are also very strongly associated with strong emotions, fear and joy and things like that. And so what we find is that, and it kind of makes sense if you think about it, but your long-term memories, you have long-term memories of things that were really, really good and positive in one way or another, or very traumatic. You're able to remember those very well because those activate these regions of your brain. When you study, we're not saying it's traumatic. It might be for you, but we're trying to activate those regions. Whereas when something traumatic happens, those regions automatically are activated, which sort of helps you sort of preserve that and remember that. Okay, so tell you what, we're going to stop there. That's the end of Bio 2 lecture. I hope everyone had a good semester. It's been the best semester. Nothing went wrong. It's just, isn't it crazy? It's kind of a crazy time. So anyway, we'll stop there. Final coming up soon. And everybody stay safe. Stay inside. Study. Think about your amygdala and your hippocampus. And we'll see you soon online one way or another.